Greeting good people. We meet once again to look at this topic which is called fertilization and pregnancy. But our focus here will be based on fertilization, the fertilization process which will be linked with the formation of multiple beds which are twins. When we look at this question, or when we look at this small concept, there are a few things that I want me and you to focus on. The first thing that we must be able to talk about, we must be able to identify the fertilization process if we are given in a diagram. So you must be able to tell that this is a fertilization process. And one of the simplest way of identifying the fertilization process is whereby you will see that the sperm cell is fusing with the egg cell. But we'll get to that part as we move along. We have to also be able to define the fertilization. What is fertilization? Then we have to also define gestation, which is pregnancy. But a word that we must familiarize ourselves is that it's gestation. We have to also talk about FREEMARTIN. We have to understand that a FREEMARTIN is a type of a twin, but you have to understand what type of a twin and how does this twin, or how do these twins develop in a cow. So we will look at that one, and then when we get into gestation, then we will also talk about placenta. So it's very important to define fertilization, define gestation, and also define FREEMARTIN. We have to continue, good people, and now get deeper and look at the process of fertilization. Say, if we say this is fertilization, yes, we have defined it, but how does the process take place? And we'll also be assisted by what? By a diagram in order to understand this process. And there's something that I want you to note when we look at that one. We will continue, good people, and describe the formation of multiple beds. We have three types of multiple beds, or three types of twins. You have to know how do each twin develop, or how do we form a monozygotic twin, how do we form a dizygotic twin, and how is a FREEMARTIN formed. So we will look at all this process, and we will be assisted by diagrams that you will see as we continue with our presentation. That's what we will check, good people. We will then look at the factors that cause or that lead to the formation of what? Of FREEMARTIN. We will not get into the pregnancy in this presentation. We will look at gestation later. Okay, fine, good people. Let's try to check what can we get here. Let's look at the process, which is fertilization. When we talk about fertilization in a simpler way, we used to say this is just the fusion. The fusion of what? The fusion of the nucleus from the ovum and the nucleus from the sperm cell to form a single cell, which is called a zygote. That's the simplest way to understand it. This is the fusion of a nucleus from the ovum and a nucleus from the sperm cell to form what? To form a zygote. That's how we have to understand it in a simpler way, whereby we say the fusion of the nuclei of an ovum with the nuclei of a sperm to form a single cell, which will be given a name and say it's a zygote. We will look at a diagram as we move along. Let's try to understand this process, good people. When you want to understand these processes, that the sperm cell will approach the egg cell. When the sperm cell approaches the egg cell, then there will be the reaction from the acrosome. Remember, the acrosome is that cape-like structure that covers what? That covers the egg cell, that is on top of what? Of the sperm cell head. Then there will be a reaction from the acrosome. And this reaction will lead to the change in the head of what? Of the sperm cell. And this reaction of the acrosome, it will cause the sperm cell to release enzymes, and these enzymes that will be released by the sperm cells, I used to give it this name and say it's hyaluronidase. What will happen is that these enzymes are capable, or these enzymes are capable of dissolving the membranes of the egg cell. Once they dissolve the membranes of the egg cell, they will then cause, or they will lead to the nucleus of the sperm cell to penetrate through the egg cell. Once the nucleus of the sperm cell has penetrated through the egg cell, then a membrane will be formed whereby it closes the egg cell. And then once it closes the egg cell, the tail of the sperm cell is being cut off. What will happen is that the nucleus of the sperm cell will then go and fuse with the nucleus of the egg cell. That's the simplest way we can understand it. Once it fuses with the egg cell nucleus, and then it will form a diploid, which is called what? Which is called a zygote. But let's look at it in this presentation. What will happen is that the acrosome reaction is a structural change of the head of the sperm. What happens? That releases enzymes. Take note of the name of the enzyme. That releases enzymes, which is called hyaluronidase. I used to pronounce it in that way to master the spelling. These enzymes will come from what? From the acrosome. They will ask you this question in 1.3, section A. What is the name of the enzyme that degenerate or that dissolve the membranes of the egg cell, which comes from the acrosome? You say hyaluronidase. What happens is that these enzymes dissolve the covering around the ovum so that the sperm cell can penetrate the wall of the ovum. Then the tail of the successful spermatozoa, because you know that there are millions of spermatozoa that will be released there, but the tail of the successful spermatozoa breaks off during the penetration, and the head of the spermatozoa fuses with the nucleus of the ovum to form what? To form a diploid zygote. Why we call it a diploid zygote? Remember that the sperm cell, or the nucleus of the sperm cell, is haploid because it was produced through meiosis. The nucleus, the chromosomes in the nucleus of the egg cell is also haploid. So when a haploid nucleus of the sperm cell and a haploid nucleus of the egg cell fuses together, they will form a diploid cell, which will be given a name and called what? A zygote. That's the simplest way to understand it. But then allow me to take you to a diagram which I believe that it will assist all of us to understand. This is the fertilization process, good people. If you try to look at it, this is your process 1, process 2, 3, 4, 5, 6, and 7. What happens is that the sperm cell is approaching the egg cell. When it approaches the egg cell, then there will be a reaction from the acrosome. When the acrosome reacts, what it does, it releases the enzyme. What do we call that enzyme? Hyaluronidase. Once it releases the enzyme, what will the enzyme do, good people? It will degenerate, as you can see here. It will dissolve the covering of the membranes of the egg cell. Once it dissolves the membranes of the egg cell, you can see that the head is now penetrating. The head is penetrating. Up until here, now the nucleus has been injected or inserted into the egg cell. Once the sperm nucleus has been inserted into the sperm cell, then it will fuse with what? With the nucleus of the egg cell. So there's a sperm cell nucleus and an egg cell nucleus. They come and they fuse together. When they fuse together, what happens is that this tail will break off, and when it breaks off, the egg cell will again form this layer. It forms the layer to prevent other spermatozoa to enter the egg cell. So once it has been able to form the layer again, so it means there are no other sperm cells that will enter the egg cell. Let's come back to the sperm cell nucleus and the egg cell nucleus. Here they are. Then they will fuse together to form, now it's this one, we said it's haploid. It's haploid. When they fuse together, they form what? A diploid. Hence we have what? A zygote now. Then the zygote will then develop to an embryo, from an embryo to a fetus, which is the part that we'll talk about when we look at what? At gestation. So in this case now, we have what? Fertilization. But make sure that you master these 2 or 3 things. Know the definition of fertilization. The fusion of the egg cell nuclei and the sperm cell nuclei to form a single cell called zygote, or a double cell called zygote, a diploid cell called zygote. How does the process take place? The acrosome will release enzymes given a name, said hyaluronidase. These enzymes will dissolve the covering of the ovum as it allows the egg cell, the nucleus of the sperm cell to penetrate or to enter the egg cell. And then it fuses with the nucleus of the egg cell to form what? To form a zygote. That's the simplest way, good people, to understand this process. That's the simplest way. Okay, fine. So the whole process is called fertilization. I say to you, we will look at fertilization and the process. After that, we will then look at the formation of multiple beds. Let's get to that part, good people. When we look at the formation of multiple beds, we are looking at the formation of twins. We want to know, how are twins formed? In a farm animal, which is a cow that we look in this as an example here. The first thing that I want you to understand, I want you to know that we have 3 types of twins that we study about here in our grade or in our level of studying. We have 3 types of twins. One, we have a twin which is given a name and said it's monozygotic twins. You can also call it identical twins. We also have the second one called dizygotic twins. They sometimes give it a name and say it's fraternal twin. Then we have the last one, or the third one, which is called FREEMARTINism. Let's look at how each of these 3 twins are formed. We said we have monozygotic twins, and take note of the other name, identical. Once we say identical, there is a part that I will talk about as we move along, which will talk about the physical appearance or the phenotype of these twins, whereby I will say they are identical. We also talk about dizygotic twins, fraternal twins. We move on and talk about FREEMARTINism. The most important thing, my good people, let us understand how each of these 3 twins are formed. Okay, we talk about monozygotic twins. The first thing that I want you to understand, when we talk about mono, the word mono, it simply means one. This is the key word that you can use here. It simply means one. So there are 3 things that I want you to note here. When we talk about a monozygotic twin, mono, one. The zygotic is taken from zygote. Mono is taken from one. The zygotic is taken from zygote. So what does this mean, good people? This simply means that this is a twin, or these are twins that develops from the fusion of one sperm cell and one egg cell. So what does it mean? It means there will be one sperm cell that will fuse with one egg cell, and then they will form what? A zygote. Take note of this part. This zygote, it will split, it will form 2 identical embryos. The zygote split to 2 identical embryos, which will be our twins. But this is something that I want you to note. When this one sperm cell fuses with what? With the egg cell. To form what? To form one zygote. This zygote, it has to divide. It has to go under division. The division that this zygote goes through to form the two identical twins, we give it a name and say it's cleavage, mitotic division. It's cleavage, mitotic division. 3 or 2 things I want you to note. When we look at monozygotic twins, there will be one sperm cell from the mono means one. One sperm cell that fuses with one egg cell to form a zygote. The zygote will develop to what? To an embryo. But how does the zygote develop? The zygote has to undergo a division. What is the name of that division that the zygote undergo? We give it a name and say it's cleavage mitotic division, which will lead to the formation of the two monozygotic twins or the identical twins. Simple. Let's try to look at it, good people. We say the monozygotic twin develop from a single ovum fertilized by a single sperm cell to form what? To form a zygote. A single ovum that is fertilized by a single sperm cell to form what? To form a zygote. Then what will happen, good people, is that the zygote split into two identical embryos. Monozygotic twins, they share the same placenta but develop from different amniotic sac. These two bullets are very crucial. These monozygotic twins, when you look at their physical appearance, their phenotype, we say they are identical. When you look at their gender, their sex, we say they have the same gender. They will ask you this question whenever they ask you about formation of twins. What you must note is that one egg cell fuses with one sperm cell to form a zygote. The zygote has to develop, ending up to have the twins, the identical twins. How does the zygote divide? It will divide it through a type of division that we call cleavage mitotic division. Cleavage mitotic division. Now we have the twins. If they ask you about the physical appearance or the phenotype of the twins, you will say they are identical. They ask you about the sex, you say they have the same sex. If we can go deeper and check why they are identical, is because, take note of this part, why the twins are identical. Hence we say, when I say take note of this word whereby we say identical, because the word that says identical, it simply means that they are the same. As a result, we say even the physical appearance or phenotypically they are identical. Okay, fine. The twins are the same or they are identical in terms of the phenotype because these two twins will share the same genes the same genes of one sperm cell and the same genes of one egg cell. So these two twins are identical because they share the same heredity or the same genes of the two nucleus from the sperm cell and the egg cell. So as a result, since they share the same genes, it means when you look at them physically, so they will be identical. Coming to the issue of gender, they will have the same gender because if the sperm cell contributes the X chromosome, it means both the twins will be female. But if the sperm cell contributes the Y chromosome, they will be males. We will get into this one when we look at sex determination, when we talk about genetics in paper 2. Okay, fine. That's what I wanted you to note on this one. Let's look at a diagram that will assist us to understand everything. Here we are, formation of multiple pairs. We are still looking at monozygotic. I said to you, my good people, mono, this is your monozygotic. This is your monozygotic. Mono, there will be one sperm cell. The one sperm cell will fuse with one ovum. This ovum will form what? A zygote. Allow me to say sperm. Allow me to say ovum. They fuse to form what? A zygote. The zygote will develop up until they form what? Identical twins. Remember, we said this zygote has to divide. When they divide, they will divide it to form the two identical twins. Which type of division do they divide? I said to you, we will give it a name and say it's cleavage, mitotic division. It's cleavage, mitotic division. Physical appearance, phenotype, we said they are identical. Hence, we give them a name and say these are identical twins. Gender, we said in terms of gender, they are the same. If they are both male, they will be male. If they are both female, they will be females. Then you can try to understand why we say physical they are the same, why we say in terms of gender they are the same. But the one that they like to ask is that why they have the same phenotype. It's because they share the same genes from one sperm cell and one egg cell. That's the simplest way of understanding this. So when they give you such a diagram, just count the number of sperm cells. Once you see one sperm cell, it means mono. One egg cell, mono. So it's monozygotic. Allow me to talk about the dizygotic. When we talk about the dizygotic, good people, I want you to understand that di means two. So if we say di means two, take note of the other name and say we can call it a fraternal twins. If we say di means two, what does this mean? If you can try to understand it using the number of sperm cells and the number of ova, it means here, in this case, there will be two spermatozoa that will fertilize two different ova. In dizygotic twins, two different spermatozoa fertilizes two different ova to form what? Zygotes. Then the zygote will end up having what? Giving us what? Fraternal twins. We will talk about the gender and the genotype. Okay, fine. Let's try to look at it. But understand that the di means two. Okay. Developed from two different ova fertilized by two different spermatozoa. So it means, good people, in this case, you will have what? A sperm cell and another sperm cell. How many are they? There are two. They will fertilize how many ova? Two. So it means you will have what? Two ova. Then that will give what? A zygote. That will give what? A zygote. Which will end up forming what? Fraternal twins. Okay, fine. The zygote, the dizygotic twins each have their own placenta and amniotic sac. Okay, fine. They are not genetically identical. Why they are not genetically identical? It's because these two twins will develop or will come from different sperm cells. So these different sperm cells, it means they don't have the same genes. They have different genes. So as a result, when you look at the phenotype, they are different. So the reason they are not the same in terms of the phenotype, the two twins comes from two different sperm cells and two different egg cells. Okay, in terms of gender, they may be the same, they may not be the same. So in this case, we say they are not always of the same gender. So sometimes you find that they are both males, sometimes you find that they are not of the same gender, whereby you have a male and a female. Let's look at a diagram. This is what I said to you. You have what? One sperm cell, the second sperm cell. You have an egg cell and the other egg cell. As a result, we say the di means 2. So it means there will be 2 spermatozoa that will fertilize, or that will fertilize 2 different egg cells to form the zygote, to form 2 zygotes, which will develop to form what? Fraternal twins or dizygotic twins. In terms of the phenotype, they are not the same. In terms of the gender, they are not always the same. Take note of not always the same. So when you get such a question, they say identify this type of twin. Look at the number of sperm cells and the number of egg cells. In this case, there are 2. So what does that mean? It means it's a dizygotic twin. When they want you to provide a reason, then you come and say it's because there are 2 different spermatozoa that fertilizes 2 different sperm cell, two different egg cell, pardon me on that one, to form 2 zygotes. Simple, 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 simple. Okay, fine. Let's look at this last one. When we look at the FREEMARTIN, it's simple. They will ask you, what is a FREEMARTIN? A FREEMARTIN, good people, we say this is a sterile. I like the term sterile. This is a sterile female calf that will develop in the same uterus with a normal male calf. So a FREEMARTIN, it is a twin that is a female, but this female twin, it is sterile. When you say sterile, we say it is permanently infertile. It cannot produce offsprings. Then we have to understand why it has to be sterile. What makes it to be sterile? You will get this one in section A 1.3. A FREEMARTIN is a sterile female calf that develops in the same uterus as the male calf, as a normal male calf. Then what you must understand here is that these 2 twins, good people, they share the same placenta. If I can just provide you with a background. These twins, the male and the female, they share the same placenta. If they share the same placenta that is connected to their mother, what does that mean? It means they will share the blood and the placental fluid. So the placental fluid that can go to the female can also go to the male. That can go to the male can also go to the female. They share the blood and the placental fluid. Then, if you try to take it, it means these 2, they share the same fluids. But there's this thing that is crucial. They say, in terms of sex differentiation, which animal or which twin is quicker to differentiate whether it will be a male or female? They say it is the male. The male differentiates first or earlier than the female. What does that mean? It means the male will secrete its hormone, which is testosterone. This hormone, since they share the same placental fluid, it can be passed to the female twin. So once this hormone is passed to the female twin, what does it mean? It means the female twin will end up developing some male characteristics. Yes, it is a female, but it will develop some male characteristics. Then these male characteristics will lead to to something else, you'll find that the reproductive tract will be developed, the reproductive tract of a female calf. You will find that the ovaries will be developed. The uterus is underdeveloped. And if the uterus is underdeveloped, it means it cannot produce hormones that will maintain what? Pregnancy. And even the udder of the female twin will be underdeveloped, and it will also show some masculine behavior. Because of all those characteristics, it makes the female twin to be sterile, because it does not represent, it does not show the female characteristics, but the characteristics that it shows are the male characteristics. Understand this one. These twins, they share the same placenta. If they share the same placenta, what does it mean? It means they will share the same fluid. If they share the same fluid, then we say the male twin is the first one to differentiate its gender. If it is the first one to differentiate its gender, it means it will be the first one to show the characteristics or to release the hormone which is called testosterone. This hormone which is called testosterone will end up going to the female. Once it goes to the female, it will make the female twin to show some male characteristics. And if it shows some male characteristics, it means it will inhibit the development of some female characteristics. That's what I want you to understand on this one. This is what we're talking about. A FREEMARTIN is a sterile calf that develops in the same uterus as what? As the normal calf. They are zygotic twins that share the same placenta. Dizygotic twins that share the same placenta to the mother. This means they share blood and placental fluid. This exchange of fluids results in the mixing of antigens that are unique for sex characteristics. Sex differentiation in males takes place several days earlier than female fetus. The male hormone testosterone is passed from the male twin to the female twin. This results to the female developing some male characteristics. What will lead to? Underdevelopment of reproductive tract of the female calf, underdeveloped ovaries that cannot produce ova. So if it cannot produce ova, it means there will be no offsprings that will be produced. There will be no ova that will be fertilized by the sperm cell. Underdeveloped uterus and inability to produce hormones that will maintain pregnancy, which are whereby we talk about the progesterone. Underdeveloped udder, so it means there'd be less production of milk. It expresses some masculine characteristics. Okay, that's how I want you to understand. Let's look at this one, which will be the second last slide. Types of twins. When we look at the types of twins, we have twin set A, which is this one, B, which is this one, then C. Take note, it's A, B, then C in the middle. Okay, fine. I understand this one. Let's focus on A and B. If they say, which twin set A, what is the name of twin set A? I said to you, good people, count the number of spermatozoa that you see. How many spermatozoa that we see? It's 1, 2. How many egg cells that you see? 1, 2. So it means there are 2 different spermatozoa that fertilizes 2 different egg cells. So since there are 2 different spermatozoa that fertilizes 2 different egg cells, then this means that it is that twin which is called dizygotic, which can be given a name which is fraternal. When they want a reason, your reason is your definition in a simpler way. There are 2 different spermatozoa that fertilizes 2 different egg cells. Safe on that one. Twin set B. How many sperm cells that you see? 1. How many egg cells that you see? 1. What does that mean? It means there is 1 sperm cell that fertilizes 1 egg cell to form a zygote. What is the name of that one? You say it's monozygotic. You are safe. You are safe. The next question you may get. So they will say identify the twin. Like it or not, they will say provide the reason. So when you provide the reason, you ask yourself, why I say this twin set egg is dizygotic? It's because there are 2 sperm cells that fertilizes 2 egg cells to form 2 zygotes. That is your reason. Why I say twin set B, it's a monozygotic. It's because there is 1 egg cell. So there is 1 sperm cell that fuses with 1 egg cell to form what? A zygote. Okay. Then remember, we said this zygote will have to divide, to undergo a certain division to form these identical twins. We said, what do we call that one, by the way? We said it's what? It's cleavage mitotic division. Don't just say cleavage division, say cleavage mitotic division. You are safe, you get a mark. Then they may say, talk about the physical appearance or the phenotype of twin set B. Same, identical or not identical? You say identical. Hence the other name of this one, we call it identical twin. So here we say it's dizygotic. Here we say it's monozygotic. Physical appearance, same. Gender, what about gender? Again, they will be the same. The gender will be the same. Okay, fine. One thing that you must note, you see, they may ask you what is process 2. If you look at process 2, good people, there is a sperm cell here that fuses with the egg cell. What do we call that process whereby the sperm cell fuses with the egg cell to form a zygote? We said it's what? It's fertilization. Think about, also take note of the enzyme that comes from the acrosome, whereby we said it's what? It's hyaluronidase. Let's come back to twin set C. You look at twin set C. There is a bull and a heifer. Bull, so it means it's a male. Heifer, it's your female. You look at the diagram. Once you look at the diagram, you're not done. There's something else that is at the bottom. You read all the information that you are given in a diagram. What are they saying here? Male hormones cause sterile female. Causes sterile female. The male hormones which we talked about, which is the testosterone. So now you ask yourself a question. There's that twin that we said it is a female. And that female is sterile, meaning it cannot produce offspring. So what do we call it? We gave it a name and say it's FREEMARTIN. You get your marks. So if they can say label A, B, C, twin set A, B, C, you will get your free marks. Talk about the reason for saying twin set A is dizygotic. Two different sperm cell fertilizes 2 different egg cells to form 2 zygotes. Twin set B, 1 sperm cell fertilizes 1 egg cell to form a zygote. Talk about the gender. Talk about the phenotype. You are safe, you get your marks. You move on to the next question. What are the factors that may lead to or that determine the formation of multiple pairs? I will not spend my time here. It will be the genetics, the genes of the animal. It can be the environmental factors. It can be the type of the breed. It can be nutrition, the food that the animal eats. So that's what you can understand. The genetics, which is fertility, the environmental factors, the breed type, and the nutrition. Simple. Simple. So you can get the definition of fertilization in section A, 1.3. You can get the the enzyme that comes from the acrosome, section A, 1.3 again, or 1.2, 1.4, sorry. You can also get the types of twins in section A. And in section B, you can get it, but make sure that you follow the things that I say they can be examined. Identify using the diagram, provide the reason, talk about the gender, talk about the phenotype, then you also talk about the division that takes place there. Once you understand it in that way, you will be safe. In terms of identifying, just count the number of sperm cells and the number of egg cells. 1 sperm cell mono, 2 sperm cells dizygotic. Then you are safe. Then you also talk about the factors that can lead to multiple births. You will be fine on that one. Then we will quickly look at the gestation period, because now we say we have the zygote. Then let's see how does the zygote develop up until we get to the fetus and having what a color to up until we get to fetus. Then later on we will look at the parturition process, which is giving birth. Okay, thank you, good people. Let's meet later.